The next speaker is Dr. IL18 Limaye. He is Associate Professor, Department of Biosciences and Bioengineering, IIT Guwahati. And his area of interest is hormone regulation of gene expression, endocrine cancers, and mechanisms of action of bioactive compounds from natural sources. He's done his PhD from Indian Institute of Science, Bangalore, and he's also a member.
Okay, good morning everybody. Am I audible?
Yes.
Yeah. So in keeping with the theme of this meeting, what I've decided to do is speak about a molecule which was isolated about 100 years back, in 1925 to be precise. It turns out that this is a furanoflavonoid and was isolated from what is called the Pongamia oil. Now Pongamia is, in most of our vernacular languages, is called Karanj, and which is why this particular compound was named Karanjin. Now if you look at our traditional systems of medicine, Pongamia oil and the different parts of this tree have been utilized for their medicinal uses. However, the traditional knowledge has still not been scrutinized with the modern methods and techniques that we have available today, and which is why I feel that there is still a lot of scope in this area in terms of wanting to unravel different kinds of chemicals and the mechanisms of action which can have potential health benefits. Now this compound Karanjin is not as popular as let's say curcumin or resveratrol or other furanoflavonoids, but nonetheless there are investigating groups in India as well as abroad who have significantly contributed to the current understanding of what this molecule does to different kinds of cells. And what I have just summarized in this slide is the current understanding of the cell biological effects of Karanjin. But what motivated us to make Karanjin as a part of our research activity are two publications which I will just briefly discuss. One was a publication which showed that Karanjin has the ability to induce cell cycle arrest and apoptosis, and for that purpose they use cell lines like HepG2, HL60 and A549. And I would like you to look at the IC50 values of this compound in these free cell lines. It is somewhere in the order of 10 micromolar to around 40 micromolar. Another publication from India itself, it essentially demonstrated the same idea, but what was remarkable about this paper is that it brought all the different kinds of components that we know of and different kinds of phenomena within the ambit of Karanjin's mechanism of action. For example, let's say P53, P21, NF-kappa B, DNA damage, caspase cascades and whatnot. But here again, I would like you to note something. Say for instance, in a cell line like some of these cell lines, I don't know whether it is visible over here, the IC50, if you look at it, they are extremely high. And that gets you into trouble because you run into a debate as to whether such high concentrations can be achieved in cells where you want Karanjin to act. So in short, whether these concentrations of Karanjin are pharmacologically relevant. But we have decided to steer clear from this debate, but then go on to ask a simple question as to, for all the cell biological effects that you know of today with regards to Karanjin, what are their genomic correlates? And this is what we believe has not been addressed in the literature as much as Karanjin's mechanism of action is concerned. So we embarked upon a transcriptomic study to look at Karanjin modulated genes using the MCF7 breast cancer cells. I may want to tell you later why we chose this cell line. And for that purpose, what we did is we used MCF7 cells, concentration set to 10 micromolar, keeping in mind the 10 to 40 micromolar IC50 concentration. And we chose to treat the cells only for a period of 24 hours. Now what you see on the left, the viability data shows you that at 10 micromolar concentration of Karanjin, which is somewhere over here, it doesn't make any difference in terms of the viable cell count. So what it basically tells us is that whatever transcriptomic data that we collect out of this experiment, our inferences are not going to be kind of influenced by some of the secondary effects or collateral effects of a system which is heavily apoptotic or heavily proliferative in nature. And so this particular data has been published, so that I am not going to get into the details of this, how RNA-seq experiment has been done. It has been done with all standard methodologies in place. However, I would like to summarize the story, what came out of this RNA-seq experiment. We found out that there are about 730 odd genes which are modulated by current gene. Roughly half of them to be upregulated and half of them to be downregulated. And as is the standard practice, we just picked up randomly some of the top modulated genes, and we have been able to show by real-time quantitative PCRs that our RNA-seq data is in agreement with what we see in quantitative PCRs. Now, when we did this experiment, we were anticipating that the kind of gene modulation that you would observe following current gene treatment should in principle reflect what is already there in the literature, meaning cell cycle arrest or apoptosis. But then we were in for a big surprise. So when we analyzed the gene expression data for the different kinds of hallmark genes which are enriched upon current gene treatment, it came to us as a surprise. I don't think you are able to see this because the table is blocking. Nonetheless, I will go ahead. That one of the hallmark gene sets which was enriched upon current gene treatment was the G2 and checkpoint genes itself. And we have looked at what the leading edge genes in this category are and went on to validate the qPCR. And what you find is there are some genes like c-MYC or for instance I think CDC25B, they are all induced upon current gene treatment. Now the manner in which current gene induces these genes goes on to suggest that it is actually causing cell cycle progression rather than cell cycle arrest or apoptosis. So this was surprise number one. The second surprise was that we also found enrichment of a category of genes which can be categorized as estrogen response early. Here again we have looked at the leading edge genes and went on to validate some of these genes, and as you can see, CD44, STC2, then TI-PAR and TFF1, they are all induced by current gene in MCF7 breast cancer cells. Now note that TFF1 is basically a classical estrogen modulated gene. So whenever we do an experiment in the lab to do estrogen treatments, the only way that we show that your estrogen treatment has worked is by showing induction of TFF1, which is also called the trefoil factor 1 or the PS2. Now taken together, the thing is that we have to note that both MCF7 as well as T47D cells, they are estrogen receptor positive cells, and they do depend on estrogen for their growth and survival. And so therefore, the simultaneous enrichment of G2 and checkpoint genes on one hand and estrogen response early genes on the other actually makes a lot of sense, particularly keeping in mind that MCF7s are BR alpha positive breast cancer cells. But then how do you reconcile this with other people's data that okay, it is actually a pro-apoptotic, and even our own 24-hour data where we say that okay, there is not much is happening as far as the cell viability is concerned. So we asked a question, what will happen if you were to stick to 10 micromolar current gene concentration but then expose the cells for a longer period of time, let's say about 120 hours. And here again we are a surprise. And what we saw here is that in fact in MCF7 cells you could actually get the cells to proliferate better at concentration as low as 10 nanomolar. And even in T47D cells that happens at concentration even lesser, in the order of 1 nanomolar. Now if you ask where is 10 micromolar concentration? So if you look at the 10 micromolar concentration data, you see that the viability is somewhere in between. It is definitely significantly less than what you see in the control cells, but then let us not forget that the cells have actually grown. The red dots are the baseline viability, and nonetheless the cells have actually grown over a 120-hour period. And so what we believe is that when you collect data on 10 micromolar concentration at 24 hours, you are basically capturing a system which is somewhere in between. It does seem to have a hangover of a proliferative pathway, but then if you were to continue to culture it at higher concentration, you would actually end up seeing cell death. One other interesting thing that we saw is that the current gene modulated genes in MCF7 cells overlapped very well with those which are modulated by estrogen itself, and for that matter the ones which are modulated by tamoxifen. And the color heat maps on the right hand side will basically convince you that if you look at the commonly modulated genes, the kind of regulation that you see with estrogen and current gene can be of both ways. Either both caranjin and estrogen can modulate the gene in a similar direction or in the opposite direction. And we also found that there seems to be a reasonable correlation between the fold changes that you see following estrogen treatment as compared to what you see following caranjin treatment. So all these data are compelling evidence to suggest and ask a question, is caranjin a molecule which can produce partial estrogen-like effects in breast cancer cells? Or to be even more direct, do we have enough evidences to conjecture that caranjin could actually be binding to estrogen receptor alpha and producing effects at least partially in that manner in breast cancer cells? So what we have done is we have actually embarked upon experiments to really figure out whether caranjin can physically bind to ER alpha. But till now we have not been successful, primarily because of the fact that purifying the recombinant ER alpha ligand binding domain has been a real great challenge for us. Every time we use different protocols, it ends up into the pellet fraction, and so we are not able to purify it. But while we keep continuing to work on that direction, it was quite heartening to find out through molecular docking and molecular dynamic simulations that it turns out that caranjin can bind to the estrogen receptor alpha ligand binding pocket in a manner which is very similar to the way estrogen would bind, particularly if you look at the hydrogen bonding with the residues which are present in the ligand binding pocket, particularly the histidine 524, which seems to be very important as far as the ER alpha ligand binding domain is concerned. But then we started to ask a question, well, we may not be having any biophysical data, but can we do some experiments that would suggest that ER alpha is involved? So for instance, what will caranjin do to ER alpha? Would it be very similar to what estrogen does to ER alpha? And that's what we started to ask. So one of the things that estrogen does to ER alpha is it increases the turnover of ER alpha protein in MCF7 cells. And it is very typical to observe ER alpha levels going down upon treatment with estrogen. And in fact, that's precisely what we see with caranjin. If you were to treat MCF7 cells with caranjin, ER alpha goes down. Then the second question that we asked was that if caranjin is modulating gene expression via ER alpha, can you neutralize that effect by doing an ER alpha knockdown experiment? So one of my students in the lab has already shown that cystatin A is an ER estrogen signaling target where estrogen can suppress the expression of cystatin A, and that suppression requires ER alpha to go and bind to the regulatory sequences in the locus of cystatin A. So as you can see over here, what you can see is that caranjin by itself can downmodulate the expression of cystatin A, but if you were to do an ER alpha knockdown, this downmodulation is lost. Similarly, the downmodulation of ADAMTS19, which is another estrogen suppressed gene, can be mimicked by caranjin, but the downmodulation can be blocked by ER alpha. I talked to you about the classical estrogen regulated gene TFF1. Now we do see an induction of TFF1 with caranjin, and that induction can yet again be blocked by ER alpha knockdown. And to top it up, we could also demonstrate using chromatin immunoprecipitation assays that while caranjin is able to modulate the expression of all these estrogen regulated genes, we could also demonstrate that ER alpha actually goes and binds to the estrogen responsive elements in all the three estrogen regulated gene loci that I have spoken to you about. So all these results finally get me to conclude that the caranjin's transcriptomic footprint is much broader than what we expected. The surprise was enrichment of G2M checkpoint genes and estrogen response early genes. While at the same time we have data that high concentrations of caranjin can induce cell cycle arrest and apoptosis, what we have also found out that at low concentrations it can actually promote the proliferation of MCF7 breast cancer cells, and that basically red flags our notion that caranjin is an anti-cancer or a potential anti-cancer agent. I think we need to look at the concentration dependence far more critically before we are able to draw some conclusions on this. So the question is, where do we go from now here? So in the next couple of years we would be busy in trying to figure out exactly the opposite of what is being said in the literature. The question is that because caranjin cannot proliferate the MCF7 cells as good as estrogen, but yet has partial estrogenic activity, could you try and use this compound in something like hormone replacement therapy? So I'll just end this talk with that. Finally, thanking some of the funding agencies. This particular project has been funded by DST SERB, and the last two students in this photograph have been the ones who have crafted out these experiments and interpreted the results. I'm happy that Akshita, who has done the entire data analysis for the next generation sequencing, is right now sitting over here with us. And since I think I have saved a little bit of time, I would be happy to answer any questions if you have any. Thank you.
Hi, very nice talk. I have only one question. For proliferation measurement, what assay you used?
Yeah, so we began by doing MTT assays,
Okay.
right? But the problem with MTT assays is that you have to have a whole bunch of 96 well plates to take the 0-hour data, right? Because we want to know from your beginning whether the cells are at all proliferated or not. Because I would only say that a compound is heavily apoptotic if my cell numbers decrease. Right?
Yeah.
So what we have done is we have chosen to do triple blue staining assays the hard way out, but probably it has given us much more insights than simply doing your MTTs.
Okay. Because sometime MTT assay lead to something else.
No,
And
they have
that's
stopped.
why we had
No.
doing MTT and BRDU together.
So,
Just to validate
so
results.
for critical experiments, we have stopped doing MTT assays in the lab.
I have one question here.
Yeah, thank you.
If you expose this MCF7 with 4-hydroxytamoxifen extensively, develop resistance,
Okay.
and then use your current gene, what will happen?
No, that's a very interesting question. We have actually not really begun to think in that direction. But sometime back we did think about a challenge experiment. Like say for example, you have current gene but you challenge with tamoxifen and the vice versa. That will actually give us clues whether they are competing with the estrogen response, I mean the ER alpha ligand bonding or not. That definitely.
Just check whether this molecule can overcome that resistance of these cells.
Exactly. So we
That
had
is a
plans
million-dollar
of
question.
generating tamoxifen resistant cells,
Right.
but we have still not been successful. I was given to understand that resistant cells are available with ATCC. So probably we would take that route in an event that we are not able to generate it ourselves.
It is not difficult to develop resistant
Right.
cells, and also is available within country.
Yeah.
Yes, sir.
In breast cancer, main problem is ER negative cells. So it won't work there.
No, it doesn't do anything to the viability of MDMB231 cells. It doesn't do anything to MDMB468 or sorry, MDMB453, which are also ER negative breast cancer cell lines. So our typical observation is that the ER negative breast cancer cells are not touched. So we do see this phenomenon of inducing the viability or viable cells in MCF7 and T47D6, but we don't see that in MDMB231.
With this dose only, 10 micromolar dose.
Yeah, 10 micromolar dose. In fact, we have looked at all doses, right from 1 nanomolar to 50 micromolar.
Did you find the same result with this T470 and MCF7?
Yeah. So in fact, because of time I have not presented it, we have done the transcriptomic analysis of T47D also.
Same?
It overlaps with MCF7, but there are differences because these are phenotypically different cell lines, although they are ER alpha positive.
And also some genetically different.
Yes.
That's why I was wondering.
Yeah. But it does overlap, but there are differences too.
It was nice presentation. I saw previous paper they mentioned about FT2 cell line. It is liver cancer cell line. What may be the effect of this compound on liver cancer?
See, I'll tell you, the moment you cross 40 or 50 micromolar, I'm 100% sure that the cells will apoptose. But the problem is that you run into that argument whether that is pharmacologically relevant concentration or not. Will we ever be able to achieve that high concentration to be able to see anti-cancer effect in vivo? I'm still not sure. So right now we are steering out of that debate and then working out the estrogen receptor angle to the mechanism of action of current gen.
Thank you, Dr. Limaye.